Hello, my name is Jude Campbell. I'm excited to present my research with you guys today over the aryleidine hydroxynyl thiazole analogs induce apoptosis in multiple cancer cell lines and exhibit the potential to be novel anti-cancer agents. Next slide, please. So our research is going to focus on anti-cancer drug development, where we are going to investigate anti-cancer activity in newly synthesized compounds. So chemotherapy drugs target various stages of cell proliferation pathway inhibiting the growth of cancer cells. So cancer is a disease caused by uncontrolled cell division. So when a normal cell undergoes DNA damage, it's going to cause programmed cell death or apoptosis, while in a cancer cell, it will continuously divide. So why do we need more anti-cancer agents? Click next, please. So anti-cancer agents have a high cytotoxicity levels causing adverse side effects. So having new therapies with low cytotoxicity and high specificity will be beneficial for the patient. And also in undergoing chemotherapy, you are going to have drug resistance as a problem. So having a broader range of chemotherapy agents would be more beneficial as well. Cancer is also very prevalent, being one of the most leading deaths worldwide, so that is also an issue. So observing this diagram, you're going to see that creating an anti-cancer agent takes several steps, and we are going to be focusing on step 1. Click next, please. So discovery and screening. First, we're going to get a library of compounds. We're going to screen these compounds for cytotoxic effects, test for their mechanisms of cell death, and then in further research, investigate the molecular target of the compound. And as you can see, this is the foundation of drug discovery, and alone takes 3 to 6 years. Next slide. Here are the analogs that we're going to be testing on multiple cancer cell lines. As you can see, they're very similar in structure, but they will vary in the cancer cells with their anti-cancer activity. Next, please. So our collaborators in India, they researched through literature and found that thiazole derivatives showed anti-cancer cell proliferation activity by inhibiting DNA synthesis. So they created 20 newly synthesized aryleidine hydroxynyl analogs, next please, where we will test these effects on multiple cancer cells. Next please. So using an MTT assay, which is a colorimetric assay, next please. The product we will be observing will show us the relative percentage of viable cancer cells. So here, after we treat our cells, and we are going to observe the absorbance levels through our spectrophotometer. So using PBS, you see 100% cancer cell survival rate, and then in doxorubicin, which is a known chemotherapy drug, and you'll see it's under 20% cancer cell survival. So we found that PS371, 376, and 379 showed a consistent, they inhibited cell proliferation greater than 50% in multiple cancer cell lines. Next, please. So here is a table showing all of the compounds as well as all of the cancer cell lines we treated them on. And you will see PS371, PS376, and 379 here showed a decrease of cancer cell survival throughout multiple cancer cell lines. So breast cancer cells, MCF7, BT474, are leukemia cells, the MOL4 cells, lung cancer cells, A549, pancreatic cancer cells, BXPC3. Next, please. So to observe the cell morphology, so how our compounds are physically changing our cancer cells. So here in PBS and PS373, which didn't really cause cancer cell death that we found through compound screening, we see that they're adhered to the plate. So MCF7 is a breast cancer cell, and they are an adherent cell line. So that's what we are expecting to see. But when treated with doxorubicin and our compounds 371, 376, and 379, we see an abnormal morphology. So they're not adhered to the plate anymore, and they show a rounded membrane structure. Next, please. And then next, please. So here we're going to observe another mechanism of cell death, so apoptosis, and we're going to use a colorimetric assay using caspase 3 and 7 enzyme. So caspase 3 and 7 is a protease, so they degrade cellular components. So an increase of these caspase proteins suggests programmed cell death or apoptosis. So we can see this in graph A, that camptothecin, which is also a chemotherapy drug, has high levels of caspase 3 and 7, as PBS, which is our negative control, will show low amounts of that caspase 3 and 7 activity. Next. And here we are going to observe at 180 minutes, and we see that PBS, it has 1, while Campto has 2.59, and our compounds show an increase compared to our PBS, suggesting that they have an increase of caspase 3 and 7 activity when treated with our compounds. So that further proves that these are undergoing apoptosis. And this is specifically in MOLT4 cells or leukemia cells. Next, please. And this is my favorite, the little pretty pictures. We have the mitochondrial membrane potential. So using a JC10 stain, it can be shown in multiple forms. So the red is going to show the aggregated form in the mitochondria, while the green is its monomeric state. So JC10 aggregates in the mitochondria at a healthy voltage. So you'll see in the PS365 and PBS, you see a high fluorescence of red, meaning that aggregated JC10 was able to go in there and the cells are healthy. And while in camptothecin, PS371, 376, and 379 show a decrease or a decrease in that red or JC10, indicating that they are undergoing apoptosis, as mitochondrial dysregulation is suggested to induce apoptosis. So this was also shown in MOLT4 cells. Next, please. So overall, our newly synthesized hydroxyl thiazole analogs, PS371, PS376, and PS379 showed inhibited cell proliferation in multiple cancer cell lines. We showed that they disrupted plasma integrity and that they induce apoptosis by increasing caspase 3 and 7 activity, and showed a loss of mitochondrial membrane potential. So our compounds have the potential to be new chemotherapy drugs. However, since this is the foundation of anti-cancer drug development, further research would be needed to create these, to make them anti-cancer agents. Next, please. So in our future work, we want to look at apoptotic genes in the cell cycle. So using RT-PCR to test for mRNA expression levels of known apoptotic genes, we can, specifically, we're going to look at the BCL2, BAX, PARP2, and Site-C, and then using flow cytometry to test for apoptosis and cell cycle analysis. So where in that cell are our compounds stopping or inhibiting DNA synthesis? So here we can observe it further. Next slide. And I would just like to thank everyone here, and I would like to take any questions. Thank you for listening.